Hi everyone, welcome to Pharmagist. Myself Dr. Haimavati. I hope you like the meme. It depicts the expectations versus reality. So in molecular dynamics, scientists expect an accurate depiction of reality using molecular dynamics. But in reality, we get a poor model due to overfitting of parameters and several numerical approximations. I hope you understood what does the funny meme represent and what are the different calculations. Since molecular dynamics is a theoretical subject, it involves a lot of theory and new approximations and calculations. So it is a bit boring and dry subject. So that's why I felt including a smile on your face makes the topic more interesting. Now coming to molecular dynamics, it is a computer simulation method for studying the physical movements of atoms and molecules. So computer simulation means I have already explained it in my previous videos. So simulation means representation of the molecules in the computer or the system. So why do we want to simulate? Because we want to understand how the proteins behave in the real-time environment. So in order to create the real-time environment, we need to include a lot of approximations and parameters. So what these proteins and biological macromolecules experience in reality. So they experience the fluid conditions, the water environment. They are continuously surrounded by water and different ions and electrostatic charges. So all these we know in theory. So these all these parameters, that is the environment of the natural the protein which experiences in its nature, we have to represent it in the computer. So this involves a lot of things to be included. So for molecular dynamics, it actually tries to calculate the motion of the molecule. So we want to understand what happens to the molecule after a certain period of time. So we know that all the living things, so every living system or the living proteins or molecules, they are in continuous movement, they are in continuous motion. So after certain period of time, what happens to the protein or what happens to the molecule? So we are interested to understand what happens to the protein after a certain period of time. So molecular dynamics actually depicts this. So in molecular dynamics, we try to understand what happens to the protein after a certain period of time, or what happens to the protein ligand complex after a certain point of time. How do they behave? So what are the different changes in conformation taking place for the molecule, how the interactions change from one residue to another, and what is the energy state of the molecule after certain period of time, whether the energy increases or the energy decreases or the molecule remains stable. So all these parameters we can study using molecular dynamics. So it is a computational method which calculates the time dependent behavior of a molecular system. So in this, after performing molecular dynamic study, after giving various inputs, we get a lot of trajectories, and these trajectories are obtained by numerically solving the Newton's laws of motions. So this entire molecular dynamics calculations, they depend on Newton's laws of motion. So all the interacting particles and the forces, the potential energies are calculated using these interatomic potentials or molecular mechanics force fields. So this method is helpful to study the structure function relationships, disease pathways, and in drug design. Now, what is the difficulty to learn and understand molecular dynamics? So, in order to become an expert in molecular dynamics or do a molecular dynamics study well, you need to be familiar with a lot of subjects like physics, mathematics, computer programming, and you need to have some knowledge about the command line environment. So the different commands which are used in Linux and Python, you need to be familiar with them. So basically science students, they are afraid of maths and programming. So it is a bit of challenging task for the science students. But with practice and with certain hard work, we can do it. So you need to be familiar with the different commands and options and the file formats of the chosen simulation software. So molecular dynamics, different developers have given different softwares like Amber, Chromax, Desmond, etc. So all these different softwares, they have different file formats and commands. So every software is different from another. None of them have the same commands or the same inputs and outputs. So in order to understand, so you have to be perfect with at least one software. You cannot become an expert in all the softwares at the same time. It needs a lot of effort. So that is the problem to do molecular dynamics. And it also involves a lot of input files and a lot of analysis. So what are those things we will be learning? What are the requirements for MD simulation? So in order to do a molecular dynamic study, you need a starting structure for the molecule. Then you need the temperature, step size, and length of the simulation or run. And the input files that are needed to run the simulation. You need a PDB file, that is the protein data bank file, a protein structure file, a topology file, parameter files, and the trajectory files, configuration file, and the standard output or the log file. Now coming to what is a starting structure for the molecule. So in order to run a simulation, we need a good structure which is obtained from NMR or X-ray crystallography. So all these protein structures, they are available in the protein data bank. So you have to select a protein structure which is having a good resolution, and after that you have to download that protein structure and minimize the structure. So in order to check whether it has the correct bond lengths, bond angles, it is not too much strained or too much distorted structure. It should have a clean geometry. If you take a strained structure, it leads to errors in the simulation run, and you cannot come into proper conclusions. So in order to start and make a good study, you need a good starting structure with a clean geometry. Next is the temperature. So the temperature is the amount of energy the structure is given. So each atom should receive the same amount of energy during the run. And we need to check it after each and every step whether the energy is uniform or not. And errors in this leads to errors in numerical solutions of Newtonian solutions, and that leads to very much distorted structures. If you get a distorted structure, you cannot come into proper solution. Next one is the step size. So in this, the step size is chosen generally 1 femtosecond or 2 femtosecond. 1 femtosecond means 33,000 centimeter inverse. So what we are saying from the beginning, that molecular dynamics is the study of movement of molecules. So whenever we are saying that it is the movement of molecule, so what is the fastest thing that can happen to a molecule? So after the next second, so the fastest thing that can happen is an electronic transition. But molecular dynamics is a molecular mechanics branch. So what happens in molecular mechanics? We ignore the electrons. So we cannot take the electronic transition into consideration. So after the electronic transition, the next thing that we can think of is a mechanical change to a molecule. So what is the mechanical change that can happen to a molecule? For example, consider the IR spectrum of a molecule. So in IR spectrum, so each peak in the IR spectrum. So you know, IR spectrum, we get different peaks like this. So each peak in this IR spectrum, it corresponds to a movement of the molecule. It can be a carbon hydrogen movement or the double bond or single bond movement. So the fastest mechanical change that can happen to a molecule is a change that is taking place in a, that means the appearance of peak in a molecule is a mechanical change that can happen in a molecule. So the shortest period of oscillation in an IR spectrum is 10 femtoseconds, that is 3300 centimeter inverse. So therefore the time step chosen for MD is 1 femtosecond. So the shortest thing for 10 femtoseconds, it is 33,000 centimeter inverse. Therefore this molecular dynamics calculates many structures for each oscillation of a carbon hydrogen bond. So we get many structures. So for every movement a number of structures, that means a number of conformations are obtained. So the number of conformations increases the amount of calculations to be done. So in order to speed up the calculations, we have to constrain the bonds or the bond length. So you can sometimes we can constrain the carbon hydrogen bond lengths. So the constraining the bond length, it does not affect the movement of the structure. And we cannot constrain the angles. If we constrain the angles, it leads to inhibition of torsional strains. Therefore, in order to speed up the calculations, we can constrain the backbones, like carbon-carbon backbone in case of proteins, and in case of big molecules we can constrain different bond lengths. So this leads to a reasonable approximation, so the calculation runs faster. If we want, we can use a larger time steps. So generally 1 femtosecond or 2 femtosecond time steps are used in molecular dynamics. Now length of the simulation. So how long you can run the MD simulation? So the answer is as long as it is possible. So how much ever we can make the, according to our computational probability or the computational capacity, we can run the simulation for nanoseconds to millisecond to microsecond scale. So the first MD run, so here I want to mention the first molecular dynamics run was carried out by the scientists McCammon, Gellim and Karplus in 1977. Here they simulated the movement of a protein called bovine pancreatic trypsin inhibitor for a time period of 8.8 picoseconds. So they selected this protein because it was a small protein having 58 amino acids and a good X-ray structure was available. In this they included only 4 molecules which were visible in the X-ray structure. So the study, they revealed a rich variety of motional phenomena, and the protein was not rigid, so and it was stable without losing its secondary and tertiary structure. So that was the thing they concluded at that time. And by 1990 the computers had become more powerful, so that the another scientists, Karplus and Petsko, they carried out 100 picosecond simulations and said it was sufficient time to study a protein at that time, that means in 1990s. But in the present day scenario, the longest published simulation as of today is 1.112 millisecond simulation of NTL9 at 355 Kelvin using Anton. That means we are still talking about milliseconds. So generally in most of the published papers, we can see the simulations carried out from 1 nanosecond to 100 nanoseconds, I think highest. And but the highest which I have seen through Google is, I search for Google to see what is the longest simulation time as of now. So it has given this result, that is 1.112 millisecond. So 1.112 millisecond means it's a huge amount of calculations involved. And the second one is a 1.073 millisecond simulation, and some other simulations of over 250 microseconds. Now coming to what are the different stages in MD simulation. So it involves input preparation, energy minimization, equilibration and production. So in the input preparation, we need a good starting structure for the molecule. Then we add So we take the structure, we check whether it has clean geometry and it is a minimized and stable structure or not. Then we add water molecules around the protein. So here you can see in this figure, initially this is the protein which is having the ligand with it. So it is a simple protein without any added external elements. And here it is the protein, the same protein, but the representation has changed. So all these small dots you can see, they are the water molecules which are added around this protein. So how to add this water ions, water molecules, everything we will see in our future videos. So basically the steps involve taking an good input structure, then adding the water molecules around it implicitly or explicitly, and then adding ions to neutralize the proteins. So generally the proteins are in a neutral environment, that is at pH 7. So because these are all charged atoms and molecules, they may have a positive or negative charge, or so in order to neutralize the charge, we need to add the required ions. They can be either sodium ions, chloride ions, or potassium, etc. So we have to add ions in order to neutralize the protein. And then we have to include the different periodic boundary conditions and the ensemble values NVT, NPT, or NVE. And then we need to give the temperature. So these are some of the files which are required additionally apart from those input parameters. So they are topology files. So these topology files, they have information regarding to the what is the atom mass, atom type, partial charges, connectivity, internal coordinates, residue definitions for the molecules and the proteins. And then we need parameter files. So these parameter files, they contain the information regarding the force constants, equilibrium geometries, van der Waals radii, and other data. And next one is the force fields. So we, based on the program, molecular dynamics simulation you're running on which software, it depends. So we have different force fields. We can use CharMem, GROMAX, OPLS, AMBER, etc. After taking the input structure, so the next step is energy minimization. So energy minimization is needed in order to relieve the bad contacts for the system, because we are taking the structure, we are adding water, we are adding ions and neutralizing it. So there will be some kind of high energy present in the protein and the protein ligand complex. So we need to minimize its energy. So that is why energy minimization step is carried out. So for example, as you can see in this graph, so generally the protein may have some high energy. So it may be at this level. So this may be due to the bad contacts. So we need to minimize. So the minimization causes the molecule to relax so that it becomes a stable. It has a stable energy. So the simulation, you can get different uh, energy states for the molecule. So we have to fix the backbone atom sometimes. We have to put the restraints on the protein carbon backbone, etc. So when you are putting some restraints and trying to do the equilibration, so the molecule which is at a high energy slowly relaxes and obtains different stable stages. So this is one energy minima, this is one energy minima, and this is the lowest energy minima. So our aim is to obtain this lowest energy minimum. So we carry out the equilibration process till the molecule becomes stable and is able to attain this lowest energy minima state. The next one is the production, that is the MD, actual MD run. So here we can run the simulation from picoseconds to nanosecond scale based on the computational power. And after running the production run, the final step is the analysis step. So you can do a visual analysis of the output structure. So once you obtain, after performing the MD study, once you obtain the output structure, we need to analyze the structure, whether it is whether there is a change in conformation or whether there is a change or the structure is distorted or it is stable. So whether the contacts are and the interactions between various residues are good or not, we have to visually inspect the molecule. Then we have to carry out the RMSD, RMSF and free energy perturbation, that is the MMPB, GBSA, decomposition analysis, etc. So nowadays we can carry out a number of studies using this molecular dynamics. So this is the basic concept about molecular dynamics. So till now I have explained what is molecular dynamics and what are the different stages, what are the different steps involved in it, and how to actually carry out a protein structure run. We will be seeing in the future videos. I hope you got a clear view of the molecular dynamics. If you like the video, so do not forget to like and share the video. Thanks for watching. See you in the next video.